So thank you very much for the kind introduction. So today I will be delivering a talk on innovative trials and giving you the perspective of the industry. Here is an outline of what I will be covering today. And we dive right into the topic, future of clinical trials. So clinical research has undergone a fundamental shift over the last years, already before the pandemic, but even more so since. Many traditional processes taking on new formats to increase clinical development efficiencies, such as designing trials to be more patient-centric, and R&D costs, technology, digitalization of clinical trials, as well as advanced therapies, personalized medicines have impacted clinical research. As you see here on this slide, patient centricity, new innovative designs, which is the topic of today's discussion, and broader evidence generation toolkits and digitalization have had an impact on clinical research. FPA has been involved in that journey since years now. As you see from the previous data, Europe is facing increasing competition from other regions in the race for medical excellence. FPA has taken really a serious stance from the top level to the subgroups, supporting innovative clinical trials. In March 2020, FPA's subgroup has issued a position paper on all adaptive clinical trials, and as lately in October 2021, the FPA Complex Clinical Trial Group organized a multi-stakeholder workshop with over 400 participants. So let's see more in detail why complex clinical trials are important. Biomarkers give incredible new information. Biomarker testing is a way to look for genes, proteins, and other substances that can provide information about diseases. For example, some biomarkers can affect how cancer treatment works. Biomarker testing may help to choose the most appropriate treatment for a specific patient. This is what we call personalized medicine or precision medicine. As you see here on your right, the number and granularity of cancer biomarkers has evolved significantly since the 90s. Once you incorporate a biomarker into a trial, there is less risk of bias. We will see in the next slide how the use of biomarkers in adaptive, innovative trials has revolutionized many diseases, but started with oncology. Adaptive designs are also considered more ethical because some patients have better odds of getting effective treatment and quickening the pace of treatment discovery. Let's go to another example, which is a very current one as it relates to COVID-19. Amongst all the COVID trials shown here, and its platform trials shown here on your right, I take only one example, which is the recovery trial from the University of Oxford. It's an intent to treat trial and a platform trial, as I said. This trial was designed for flexibility so investigators can make real-time decision once an arm produces data, being it thumbs up or down. The protocol put no restriction on the enrollee number nor their age. And when dexamethasone proved to be helpful, it became a standard of care. When hydroxychloroquine proved to be ineffective, the trial arm was dropped. It allowed to tackle research questions very efficiently in record time and will hopefully prepare us for a future pandemic. Let's look at the range of innovative clinical trial design. Recent advances in drug discovery and biotechnology have accelerated tremendously the detection of treatment candidates. As we saw in the previous slides, diagnostics have become more refined, leading to more precisely defined disease description, hence smaller patient populations for targeted therapies. The classical two-arm parallel group traditional randomized controlled clinical trial have thus become one of the rate-limiting factors, and more treatment trial designs have been called for. The master protocol framework remedy the problem because such trial design allow for the evaluation of investigational treatment in multiple subgroups for a study population within the same overall clinical structure. Complex, advanced, innovative design use adaptive methods such as the Bayesian methods and other clinical trial design techniques to increase efficiency. In this slide, you see several examples such as basket trials, umbrella trials, platform trials, and historical control trials. Master protocol and complex innovative designs continue to provide frameworks that hold enormous promise for engaging patients more productively, increasing the overall efficiency and improving the successful outcome. So let's go one level deeper. Despite much research in statistical literature highlighting the potential advantages of adaptive designs over traditional fixed designs, the uptake of such methods in clinical research was rather slow. One major reason for this is that the different adaptations to trial design, as well as their advantages and limitations, remain unfamiliar to a large part of the community, with lack of expertise and experience amongst clinicians, trialists, and statisticians. Hence, the experience is gradual towards the different level of adaptation. For example, broadly used are simple adaptive trial designs such as dose dropping and group sequential. Then used to a lesser extent is the selective use of advanced techniques such as master protocol designs we just saw, umbrella, basket, and platform designs, modeling and simulation used for pediatric extrapolation. Finally, true invention and trailblazing efforts include placebo substitution and historical control of phase 3 trials. If we go a level further, adaptive trial designs have their specific benefits, and I will pick only 3 examples. Adaptive randomization allows patients to be assigned to treatment arms that are more likely to be effective. We will see that in the next slides. This, in turn, may result with reduced sample size. Sample size re-estimation allows the testing of if assumptions still hold true and ensuring that the trial retains sufficient power to assess trial objectives that is key for success. Finally, adaptive enrichment design gives trial participants a better chance to receive their personalized optimal treatment, and those results in a higher response rate on the trial. Let's go into a specific example, and I picked only 2 examples. The first one here is in a very small patient population. It's umbrella trial, epoch 1 to 4, with canakinumab. Familial Mediterranean fever, mevalonate kinase deficiency, and tumor necrosis factors receptor associated periodic syndrome, TRAPS, are monogenic autoinflammatory diseases that are characterized by recurrent fever episodes such as variable skin, joint, and serosal involvement. Flares last several days or weeks and affect functional capacity and quality of life. Some of these lead to long-term complications. This trial consisted of one cohort per disease. Each cohort followed the same design here, as you can see here on the figure. The design of the trial was chosen to address the rarity of the diseases, the wide age range, and the need for a randomized controlled trial while exploiting the hypothesis of a common mediator. It also allowed the creation of a larger safety database than what could have been generated for one single disease. I have another example here, I-SPY 2, in a much larger patient population. Actually, of note, it inspired a similar setup for a COVID trial. I-SPY 2 trial of neoadjuvant treatment for locally advanced breast cancer established a new benchmark for efficiency of phase 2 trials. It is widely regarded as a pioneer of the platform trial. I-SPY 2's success continues to be a major influence of the development of next generation trial designs in oncology and beyond. So I-SPY 2 breaks from the traditional randomized clinical trial model by employing an adaptive model to increase the efficiency and minimize the numbers of participants. At consent, each of the new participants is classified in one of the 10 molecular subtypes, and each participant follows the 5 steps. The first is the adaptive randomization engine, step 1. The second is the endpoint assessed at surgery. Primary endpoint is residual cancer burden, 0, or pathological complete response as a surrogate endpoint, meaning the tumor has completely disappeared. Step 3 allows predictive probabilities of an IMP in the various subtypes are updated in real time. And finally, step 4 and 5 allow to add new IMPs and stop them if they reach futility. If we go to the next slide, I can give you an overview and summarizing the benefits of complex clinical trials. The advantages of an adaptive design is the ability to use information that was not available at the start of the trial to improve efficiency. For patients, master protocols offer personalized treatment driven by their genetic subtype and biomarker makeup. They also allow for a greater chance to be enrolled in the most effective arm. An adaptive design can provide a greater chance to detect the true effect of a product, often with a smaller sample size in a shorter time frame. So in balance, our experience showed that the efficiency and benefits far outweighs the complexities. And then we go to the next slide. However, there are still some challenges, and complex clinical trials are not one size fits all. In recent years, solutions and directions become increasingly evident. As to the challenges, further improving technical and statistical capabilities in the national competent authorities and ethics committees for reviewing complex clinical trial protocols in a timely fashion would be welcome. Likewise, sponsors have to develop the best practices to describe and justify the choice of design and key design parameters, including their assumptions. Enhancing regulatory guidance on acceptable complex clinical trial designs, analysis, and reporting would also be helpful. Finally, EMA could consider establishing a cluster with its agency peers to discuss complex clinical trial-related issues. We have seen from the workshop in October the need for a collaborative multi-stakeholder platform to facilitate interaction and learning amongst all the stakeholders. But of course, with keeping a careful line between advisory discussion and collaborative discussion, and respecting the mandates and responsibilities of each of the stakeholder groups. So there are still some practical issues in implementing complex clinical trials. These include appear with any program, but they might be even more acute with complex clinical trials. And as we are only a few days into the CTR implementation, it is too early to draw some conclusion. But areas are worth monitoring. For example, adaptation of endpoints during clinical trials requires a new CTA or modification. Seamless adaptive designs and between phases submission will require careful monitoring. The clinical research space is highly competitive, and I hope by now people realize the importance for Europe to ensure a healthy clinical research ecosystem. So, and this should be actually a common priority in the EU. The EU landscape is already a quite complex place to conduct clinical trials. There is a need to change for all stakeholders by prioritizing education activities, monitoring the impact of the clinical trial implementation on clinical trials, and maintaining and broadening the dialogue across all concerned stakeholders. In that, FPR very much welcomed the joint European Commission, EMA, and HMA initiative accelerating clinical trials in the EU. We have all a common goal to ensure patients a healthy clinical development landscape, enabling the generation of robust evidence for decision making. With that, I thank you very much.